Good morning, good afternoon. How are you, everyone? I'm Cherry Kubota at the Ohio State University. Welcome to Indoor Ag Science Cafe, October. This is the platform to communicate each other and exchange information, and scientists present research update and industry members present what's going on. So I really like to welcome. So right now we have a relatively small group of participants, but I'm probably I expect a little bit more today. Right, and then I see that many are joining now. All right, so before starting today's topical presentation, I'd like to just remind upcoming cafe series. Cafe is usually Tuesday, 11 a.m., and then the upcoming ones are also in that schedule. So today is the first one. We have a speaker from Ohio State, the Ohio State University, and then the November Cafe. It's going to be an open discussion day, November Cafe. So OptimIA is the multi-state project funded by USDA SCRI program. And then we will have most of members gathered together and answering your questions. So that's the open discussion day. In December, we will have Dr. Muraka Chira, who will be presenting modeling-based simulation of climate conditions in indoor. So this is a quite interesting area. I hope you enjoy that. And then I'm in a discussion with potential speakers for coming January and February. So I will, as soon as I know, I will let you know. So with that, today's agenda, the first part is going to be recorded one. Dr. Sanja Ilic from the Ohio State University Department of Human Science in the program of nutritional science or human nutrition. And then she's the state, let me go back again. She's a food safety state specialist, understanding agriculture. So let me stop sharing and then have Sanja to start her presentation.
Okay, I'm going to quickly share screen and then introduce myself. Thank you, Cherry, and thanks for the organizing committee for inviting me to talk to you today.
Sanja, it's in editorial. Yeah.
Okay.
Start the presentation.
Give me 1 second. Okay. Yep. Everybody sees the slides?
Great.
All right. So, Cherry, thanks for inviting me. And just as Cherry mentioned, my name is Sanja Ilic, and I am an associate professor in human nutrition. That's in the Department of Human Sciences and in College of Education in Human Ecology. But I also have an appointment in extension, and I'm a food safety state specialist with Family Consumer Sciences in College of Agriculture. So my extension appointment gives me mandates me to translate the science into practice, and my research focuses a lot on hydroponic fresh produce production. Okay, so today I'm going to be presenting some research from my lab, and also want to say that most of this research is done in collaboration with my dear colleague Melanie Louis-Ivy, who is a plant pathologist and a faculty in the Department of Plant Pathology, but her focus is also looking into co-management between human pathogens and plant pathogens. Fun fact about me: before joining the Ohio State University, where I did my PhD and postdoc working on hydroponics, I did my master's at the University of Guelph in Canada, and then worked as a food safety manager in a processing plant, leafy green processing plant, for 2 years. At that time, that was back in 2006, the outbreak with spinach just happened in the midst of us setting up the food safety practices in the plant. And it was basically prompting us to set up some firmer food safety measures to ensure that we prevent those outbreaks of human pathogen outbreaks from fresh produce. Okay, so today I'll be talking a little bit about the research that comes from my lab, but just as introduction, we know that, and I'm preaching here to the choir, but we know that hydroponics have a number of benefits to field production. So in addition to being soilless, better growth, better yield than from the field, the reduced need for the pesticides and herbicides, and the nutrient efficiency that we get from hydroponics, we also can serve consumers who are requiring year-round products. So season is long, production is long, the yield is higher. We can manipulate environment to even improve those parameters, and we can introduce automation to then further improve the efficiency, reduce labor potentially, etc. So, but I know you know all this because I'm preaching to the choir. Just to the overview of the market share by crops, they say that half of all the tomato that is sold fresh in our grocery stores is coming from the greenhouse production, hydroponic production, and most of it is hydroponic. So there's some soil and base production that is from greenhouses, but majority of that is hydroponic. And then a lot of peppers and cucumbers, increasing number of lettuce operations and leafy green operations is emerging in Ohio and nationally, and definitely requires the look at how safe we can produce, and we can grow the hydroponic production in a way that is going to be also safe for human consumption. All right, and the projected growth for the industry is really surpassing anything that you're expecting from the field, and the majority of that growth is expected in leafy greens and other crops like strawberries, microgreens, etc. So now, as I said, I'm preaching to the choir, and I know that you know that part, but what is maybe less common knowledge in hydroponic production is the food safety aspect of it. So we know that certain crops are better to be grown hydroponically than others, and the majority of ones that we do grow hydroponically are the fresh produce crops that are linked to previous outbreaks of human diseases, of foodborne diseases. So leafy greens are the top risk for foodborne outbreaks, leafy herbs as well. Strawberries have been linked, green onions have been linked, cucumbers have been linked with various human pathogen outbreaks, peppers, etc. So we do grow these crops hydroponically, and whenever we grow these crops hydroponically, there is a concern that these crops may be contaminated with human pathogens. So this concern is real, right? It's not something that we're just anticipating. It is something that has happened. So this is back in 2013, we have a major multi-state outbreak of Salmonella St. Paul infections that were linked to the cucumbers. These cucumbers were imported from Mexico, but they were grown hydroponically in Mexico. At that time, nobody died from this salmonellosis, but a number of people were affected and hospitalized. Okay, the other major outbreak that probably many of you remember is the outbreak of E. coli O104. So this is Shiga toxin producing E. coli, the killer E. coli that was linked to fenugreek sprouts in Germany that actually made almost 4,000 people sick. And out of those, almost 1,000 people developed hemolytic uremic syndrome, which is the disease that debilitates the kidneys, right? And 53 people died. And those fenugreek sprouts were grown in hydroponic. But in addition to that, if you remember that instance, that first the E. coli, the similar strain, was isolated from greenhouse-grown cucumbers and was linked to greenhouse-grown cucumbers. So it was an error at the beginning of that outbreak investigation, but it serves to show that human pathogens like deadly E. coli can contaminate greenhouse produce. Now, I have to tell you, in the study that was performed under another SCRI grant a few years back, we sampled greenhouses, tomato greenhouses, one of which is behind me here. And we have found that 33% of the greenhouses that we surveyed microbiologically had Listeria monocytogenes, which is another pathogen that causes foodborne diseases that are very serious and cause deaths. The Listeria monocytogenes was present in 1/3 of the greenhouses that we surveyed in the environment, but not on the product. And we know if some human pathogen is present in an environment, there is a possibility that the product will get contaminated. All right, so we have heard over and over again that romaine lettuce can be contaminated with E. coli, and a lot of times the source of contamination of E. coli on romaine lettuce is going to be linked to some sort of animal activity around the field. At least that has happened in the 2018 outbreak of E. coli, if you remember, when all romaine lettuce had to be recalled because there was no way to distinguish between hydroponically grown lettuce and field-grown lettuce at that point. In the meantime, some food safety recommendations and food safety guidelines were put in place to improve the traceability and to enable the hydroponic growers to distinguish their product from field growers, and in that way, ensure that their product is not going to be recalled in the case of some of those outbreaks. Okay, but at that time, there was a lot of talk about safety of hydroponic lettuce and how hydroponic lettuce should be always consumed to ensure food safety. And the hydroponic is proposed as a solution to some field-related food safety problems? Well, I'm going to be showing the research that is going to discuss and put that into context to make you understand that this is not always the case. The risks in hydroponic productions are different than the risks in the field, but they're still there. Okay, so a lot of times there is attitude in the media that hydroponic production, whether vertical or any sort of protected environment, can actually reduce the risks by reducing the exposure to sources of contamination like animals and pests. And because if we have certain setup, or if we have automation, the exposure to human handling will be reduced. Okay, and so this leads to the studies that we have performed since in the past basically 6 or 7 years. And back in 2014, we asked the industry, and at that time it was the hydroponic tomato industry, how they perceive the impact of contamination with human pathogens in terms of economic consequences to your operation. And we specifically ask for the contamination with Salmonella, E. coli, Shiga toxin producing E. coli, and Listeria monocytogenes. All the pathogens that I mentioned were linked with those outbreaks. And so the respondents, the industry respondents, tomato growers, greenhouse tomato growers, said that the impact would be critical to catastrophic, 10% of them, and then majority, unfortunately, did not know. And at that point, we're not certain in what are the hazards that may occur in hydroponic production. Okay, so for food safety risks in the field, we usually see these 4 W's as the major sources of human pathogen contamination. And these are waters, workers, so human handling, waste or different soil amendments, and wildlife. And so in the field, this is very obvious. There could be animal intrusion with by wildlife or domestic animals, then waste that comes either through runoff or is deliberately used as a fertilizer, then worker handling and hygiene, and then irrigation water that could be contaminated. But the risks in hydroponics are basically very similar, however, differently impacting the crop. So water definitely is a high risk. So water in hydroponic system has much more impact in terms of contacting the crop throughout the life cycle of crop production. The workers, depending on how intense worker intense operation is, are definitely the source of contamination. We know that 50% of all outbreaks outbreaks of foodborne disease linked to fresh produce are from norovirus. And norovirus is the virus that animals do not carry. So it comes from humans and is transmitted by humans and can be transmitted at any point of handling of the crop. And we know in certain operations like deep water systems, etc., that each plant is basically handled by a human. One human, if the transmission of norovirus is fecal oral. So a human that is carrying the virus can contaminate quite a lot of product. Pests, obviously, the pest control is critical for food safety of product produced in any controlled environment. Rodents, birds, pests, etc. Depending on how your hydroponic production looks like, you may be attracting the pests more or less. So behind me in this facility, the workflow was such that all the debris from trimming was let settle on the bottom and was not cleaned out and actually was used to breed the beneficials. So in cases like that, you have a lot of you have a higher risk of attracting the pests. Birds obviously are an issue. And in my experience, cats are a lot of times used as pest control and in some of the facilities, and definitely increase the risk of some foodborne pathogens, especially the deadly Toxoplasma gondii. Okay, so environmental condition, I mentioned already, if there is a pathogen in the environment, it is likely that that pathogen is going to end up on the product. The difference between the open field environment and the closed controlled environment is that our environment in the greenhouse is more like a processing environment than open field, right? And it's just like in a processing environment, pathogen called Listeria monocytogenes can establish itself and form biofilms. And from these biofilms, then contaminate products in gusts or systemically. All right. So let me share with you this risk assessment of tomato production, greenhouse tomato production that we conducted as a part of that SCRI grant the PI on that grant was Dr. Sally Miller from plant pathology. So during that project, we went out, developed the list of food safety practices, and we went out and surveyed, actually in-person surveyed, on-site surveyed 26 tomato greenhouses, mostly hydroponics, across the US, and some in Canada, some in Mexico, and one in Guatemala. And most of the locations you can see here on the map. The greenhouses ranged from huge operations, 100 plus acres, to very small, less than 1 acre operations that were run by one person or a couple. So for that project, we kind of constructed, we surveyed the practices, we cataloged the practices, and then ranked the risks. And we developed pretty much the flow chart of how a large-scale tomato operation looks like, and also how small-scale operation looks like. And we outlined the processes that were going on and ranked the risks of inputs and practices that could lead to food safety problems. So you can tell how large scale operation with all propagation, greenhouse production, and post harvest production looks like, and then all the inputs that like fertilizer, water, sanitation, pest management, seed management, and then waste management. The majority of the risk that was observed here was actually from the sanitation. And for the small greenhouses, the post-harvest handling was observed as the highest risk portion, because although the process of the production process went on in the greenhouse, the post-harvest treatment, like packing, sometimes even washing and handling, went on in garages, under the trees, in environments that were not very controlled for environmental contamination. So much of these practices were not very clear of what kind of impact they have for food safety, and not much. We did not know that much. We did not have much research to tell us what kind of risks are, what can we do, and what should we expect, for instance, if water in hydroponic production is contaminated with a human pathogen. And even so we went out to experts, who knew the production, the greenhouse production, and knew about human pathogens, and kind of harvested their opinion and put it in the rank. And so water management was ranked one of the top risks. And even the experts at that point did not reach consensus. And what do you do if you have a tomato plant that is an annual plant in the greenhouse, and your water is contaminated with a human pathogen? What kind of mitigation can be successful to prevent that annual crop from just having to be destroyed? So the expert opinion was ranging from, oh, you have to stop and destroy the crop, until that you do nothing. Right. And so this is just to illustrate what kind of lack of understanding of how the pathogens survive and transmit through water was there at that time. Okay, so my colleague Melanie, that I mentioned before, and I went on to study this and to answer this question: can these pathogens, Salmonella and Listeria monocytogenes, persist in the leafy green production, hydroponic production, throughout the life cycle of the plant? And we wanted to understand those risks. What are the risks, and what can we do about it? Okay, we got funding from ODA and ongoing funding to study this problem and try to solve and make some recommendations. So we did what? We actually, at the Ohio State, we have BSL-2 greenhouses, and we partnered with Crop King. We got NFT setting for lettuce production. We set it up in our greenhouses. And then we inoculated with these pathogens, Salmonella and Listeria monocytogenes, and we observed over time what happens in the water system, so in the reservoir, channels, etc. And so this is what we found for Listeria monocytogenes in the fertilizer solution. You can see on this graph the time on x-axis and the number of microorganisms that we inoculated on y-axis here. And so it's expressed in log CFU per milliliter, which are actually colony forming units on these plates. This is how we count them. And we wanted to see, does that number, let's say if we have sporadic contamination, what is going to happen with that pathogen over time during the life cycle of lettuce? And we observed that from the beginning, where we put the pathogens, all up till the lettuce was ready for harvest. Okay, and so we used 2 different levels of inoculum, and we used that because we wanted to simulate. We had an issue. We had growers approaching us, especially Crop King, the growers that were using Crop King systems. If they put the water reservoir in the ground and there is a storm or a heavy rain event, they would have that reservoir flooded from time to time. So then that contamination would be higher, and that was simulated in our experiments like this high inoculum here. So we started from these two different points, but with Listeria monocytogenes, we showed that it survived during the whole period um, of the lettuce production from when we put the seedlings in until it was time to harvest. Either way, with a small inoculum or high inoculum, it persisted over that period in both this reservoir and the channels, the lanes of NFT system. Okay, so then we repeated the same experiment with Salmonella. So same thing, different pathogens. Again, we found Salmonella did not persist in very high numbers. So the number over time from when we put in the seedlings and inoculated until the lettuce harvest, the number of microorganisms, depending on the environmental conditions, decreased all up to in the first 7 days, decreased, but remained detectable over the period of life cycle of lettuce. Okay, so we looked into as well the tissues, right? So now we showed that both Salmonella and Listeria persevered, survived, and were detectable in the water from the reservoir and the channels throughout the life cycle of the plant. Now we wanted to know what happens with actual lettuce, right? So we tested lettuce, leaves, edible portion, we tested roots, and we tested rock wool, right? And again, we counted the numbers of microorganisms over time. All right. And so what we saw is that both in high and low inoculum, the pathogen, Listeria monocytogenes, was detectable on leaves of lettuce, most likely through cross-contamination, and was detectable in high counts in that rock wool root medium, right? And so what we observed is that that rock wool root medium serves almost like a little filter and traps the bacteria in it, because we detected much higher counts in rock wool and roots than in water, right? With Salmonella, we did not detect such high counts, so the counts decreased in the first 7 days, but still were detectable depending on initial inoculum in more, in less or higher numbers, but it was detectable in the fertilizer in the tissue, both leaves, roots, and rock wool throughout the growth cycle of the plant. Again, this time in rock wool and root, we could detect higher counts of bacteria. Again, we think this is because the rock wool acts like a filter and traps the bacterial cells in it. This is of particular concern because a lot of times the lettuce can be sold as living lettuce when you're putting it with the root and that rock wool in a clamshell. This definitely is a risk. So the answer to our question basically is that yes, both pathogens that we tested in both situations, flood or sporadic, contamination, can persist in the system, in water during the period of life cycle of lettuce life cycle, and they concentrate in the area of root rockwool medium. Each time, the pathogens were detectable on the leaves of unedible portions of the plant. So contamination transfers either through cross-contamination during handling. It was practically impossible to pull the lettuce leaves from inoculated water and not have a little bit of a splash on the leaves. All right, so now the next question is, what do we do about it? Right, we now know that if a pathogen gets into the system, it is going to persist there and it is going to contaminate your lettuce, right? So what can we do about it? So to answer this question, we tested common sanitizers that are used in greenhouses. Now, going back on our surveys of hydroponic production, we did survey tomato, but what we found is that less than a quarter of all the operations actually conducted the full sanitation of their environment. And the food safety mentality in cleaning out the environment was not even there. So even if a total clean out of the greenhouse was conducted, it was conducted for the reason of suppressing plant pathogens, right? So what we found to answer the questions about the lettuce NFT system is, first of all, we tested all the sanitizers that we found in our survey that are in use in the greenhouses, and we tested them in the recommended concentrations and time and contact time parameters, right? So what we noticed off the bat when we, for our own experiment, wanted to decontaminate the systems to repeat the experiment, when we used chlorine-based sanitizer, bleach, our setup fell apart. So bits and pieces from the pump reservoir, the tubes, the connectors actually disintegrated because of the high concentration of chlorine. So, but when we use the lower concentration of chlorine for disinfection of water and equipment, you can see here what happened. So sodium hypochlorite and in two different concentrations here. So in this graph, you see different treatments that we tested, different sanitizers. Each one of these sanitizers is used commonly used in more or less commonly used in greenhouse production. And this first bar here is our positive control. This is when we put in these bacterial cells. This is how many we have. We had around 1,000, a little bit over 1,000. That is this 3 log bacteria per every square centimeter of the surface. So we wanted to show how effective these sanitizers are to disinfect the contaminated surfaces in the greenhouse. And so we tested again. We tested the tank. We tested tubes, etc., etc. This graph here shows the results on the tank. So we used just water, so without any chemical. And then we put different sanitizers like sodium hypochlorite, that is bleach, in 100 ppm, and then 200 ppm, a higher concentration. We use chlorine dioxide in different concentrations, and and then Clean Grow, Green Shield, Sanidate, Virkon, and Zerotol. Virkon is not approved to be used in food production, but we have found that a number of greenhouses is using Virkon for environmental disinfection. So we tested it. So basically, each of these treatments where you see a little bit of a bar means that these sanitizers were not able to kill all the pathogens from that surface. So, and were barely or not statistically not different from actual just water, right? And this is of particular concern when we're thinking about chlorine, because chlorine is really widely used. The bleach is widely used for disinfection of surfaces. As I said, if you increase the concentration of chlorine, you might be getting a better effect, but then your equipment is going to fall apart. So we're continuing our research in this area and finding what are some of these sanitizers like Sanidate, Zerotol that managed to reduce and kill off all the pathogens, and we couldn't detect pathogens under that. Green Shield here, we could not count the pathogens, but after enrichment, we could still detect them. So we know it wasn't effective enough. So our work on these disinfectants and disinfection of water continues. On that note, all the recommendations that we have and that stem out of our research or the research from our colleagues in other universities, or we kind of collate them and always integrate them in practical recommendations. And so last year, Melanie and I have developed the Good Agriculture Practice Guide specific to hydroponic production. We have conducted one training with Cherry Kubota and her group, and it was successful because most of Good Agriculture Practices trainings focus on field production, and we really needed something that will be very, very specific to the hydroponics. As I said, we continue to research and use research to answer the questions of how to ensure that we're producing the safe produce hydroponically. And on that note, this is just to show you how the guide, the table of contents of the guide, and how some of the information is conveyed and illustrated here on this page for food, for water risks. We also we focus on fresh produce in general, not only hydroponically, and develop resources for growers in hydroponics, but also in field production. You can find a lot of information on fresh produce production on our website, which is producesafety.osu.edu. And with that, I am acknowledging our industry partner, Crop King, for making this research possible and actually bringing some of the issues to our attention. This is our team in BSL2 greenhouses, Crop King people, our students, and Dr. Melanie Louis-Ivy, who is my collaborator on this topic. With that, I thank you, and I'll be happy to answer any questions.